Throughout the kingdoms of life, each organism is made up of a vast array of proteins. These are used to build structures and carry out the complex processes required by life. The central dogma of molecular biology states that DNA can be used to produce RNA, which can then be used to make proteins. However, some RNA is used to build structures and catalyze reactions, such as the ribosome, which interacts with other RNA molecules to produce a protein precursor. These different forms of RNA are synthesized by unique versions of RNA polymerase, which copy them from DNA. This process is known as transcription. RNA polymerase II is required to make messenger RNA, which goes on to make proteins. Polymerase I is used to synthesize the components of ribosomes. To connect polymerase II to the DNA strand, a number of transcription factors are required: TF2B, D, E, F, and H. Each plays an important role in initiating transcription, such as TF2B, which actively recruits the polymerase enzyme. Equivalent transcription factors have been identified for all RNA polymerases in eukaryotes and archaea, all except one. Despite all indications that it must exist, no protein corresponding to TF2B could be identified in polymerase I recruitment. Could this missing link be identified? Dr Joost Zomerdijk and his team wanted to find out.
We knew that there was a TF2B-like molecule involved in Pol3 transcription. There was a TF2B molecule involved in Pol2 transcription. And so it made sense, because given the conservation of all these mechanisms and processes, that there might be a similar protein involved in Pol1 transcription.
Sequence analysis could not identify a TF2B homologue in polymerase I transcription. However, structural analysis revealed TAF1B, a subunit of the SL1 complex, shared many domains with TF2B. TF2B contains a zinc ribbon domain, which facilitates polymerase II recruitment. A similar domain in TAF1B suggested it may also be involved in the recruitment step. Cyclone-like folds in TF2B interact directly with DNA. Equivalent structures in TAF1B implied a role in binding the polymerase to the DNA strand. Disparities between the proteins were also observed. Highly conserved domains in TF2B's connecting region, known as the B linker and B reader, were absent in TAF1B. Although TAF1B's connecting region is active in transcription, these domains were not observed. The TATA box binding protein is not used in polymerase I promoter binding. Instead, TAFs, such as TAF1B, fill this role. Although TAFs are present in the polymerase II assembly, they are associated with the transcription factor TF2D and not TF2B.
Despite similarities, a similar function could not be deduced based on structure. Mutagenesis experiments were devised to determine the role of TAF1B.
We thought if it's a zinc coordinating amino acids, then if we mutate those, we thought we might mutate a zinc finger and therefore not have DNA binding. But it wasn't a zinc finger, it turned out to be a slightly different variant that also is involved in coordinating zinc ions. But the mutations gave a very dramatic phenotype, and yet we couldn't pin it down to any of the obvious defects in assembly of the complex, recruitment of the polymerase, DNA binding. They were all fine. And it turned out that actually there was a defect after all these events happened. So it binds DNA fine as a properly assembled complex. It recruits the polymerase, but then there is a problem.
It was found that TAF1B was not essential for polymerase I recruitment. It appeared that its main function may occur post-recruitment. This is similar to polymerase III transcription factor, BRF1, a TF2B homologue.
Does this mean that TAF1B is related to TAF2B and BRF1? The evolution of TF2B-like transcription factors exemplifies the processes of divergent and convergent evolution. Like a human hand and a dog's paw, TF2B and BRF1 have evolved from the same predecessor, diverging to achieve their unique functions. TAF1B, like a bat's wing and a bird's wing, has evolved from a different predecessor protein, converging with BRF1 and TF2B to develop a similar function. Therefore, much is still to be learnt about TAF1B.